So what is CRISPR? CRISPR is a genome editing technology that was just recently developed in the early 2000s. CRISPR gives scientists the ability to change an organism's DNA. It allows for them to add genetic information or take it away depending on what is necessary. So how does this whole thing work? How does CRISPR work? Well, there are a couple different methods that came before CRISPR that actually helped develop CRISPR. The earliest being homologous recombination. This is, as I said, the earliest method of gene editing, and it was an exchange of homologous strands of DNA. It was first developed in the 1970s, except this type of gene editing is extraordinarily limited and inefficient. It is also incredibly inaccurate as it only worked about 1 out of 1 million times. So obviously we had to step up our game because that is not enough. The second thing that helped develop CRISPR is zinc finger nucleus. It was the beginning of the realization of CRISPR and helped scientists to develop it. Zinc finger nucleus was developed in the early 1990s. The third one, which is the most recent one that helped develop CRISPR, was TALENs, or transcription activator-like effector nucleases. This was developed in 2009 and uses proteins from the nucleus to bind to specific DNA strands. Now, the one we are talking about mainly here is CRISPR. And in its core, CRISPR is a combination of both zinc finger nucleus, or ZFC, and TALENs, or transcription activator-like effector nucleases. The full name for CRISPR is extremely long, so we call it CRISPR, but it is clustered regularly interspaced short palindromic repeats, aka CRISPR. So now, what does CRISPR do? How was it founded? Where was it discovered? So scientists actually discovered CRISPR in bacteria before they found it in humans. The bacteria captures and stores snippets of DNA from invading viruses and uses them to create things that we call CRISPR arrays. These arrays allow for the organism's cell to remember the attacker in order for the body to fight against it in a more effective way next time. If the virus attacks again, then the organism's cells produce RNA strands or RNA segments for CRISPR arrays in order to attack the virus in a more efficient way. The organism then uses protein 9 or Cas9 or a similar protein to cut apart and destroy the virus's DNA, killing the virus in the process. So how does this work for humans? How do scientists do this in a lab? Well, scientists create RNA using a short guided sequence. The sequence then binds to a specific target sequence in the DNA. The RNA also binds to the Cas9 or protein 9 as well. The Cas9 enzyme then cuts up the DNA in the targeted sequence. After this is done, scientists can use the DNA's own repair mechanisms to add and or delete different pieces of the genetic information that don't need to be there or need to be added. So what exactly can CRISPR do for us? What are some of the pros? Well, CRISPR's main thing is the fact that it can edit genes. This, in turn, can help to solve or cure genetic diseases, as if it edits a specific DNA sequence that carries a genetic disease or genetic mutation, then that person does not have to be inflicted with it. This can then allow people to live happier and healthier lives, being able to stop their suffering, especially because they didn't even have a say in whether they gained these genetic diseases. It also then helps people to live longer and contribute more to the world and even the economy, if you want to think about it in a more economical standpoint.
What are some ethical problems and/or cons that CRISPR might concern? Well, the ethical problem being focused on right now is if using CRISPR is worth possibly creating medical issues in the future. Safety concerns are one of the main issues in using this technology. Mosaicism and off-target effects are very dangerous, especially for humans. Mosaicism means that some cells carry the edit and some don't. This mutation can cause ichthyosis with confetti. It causes red scaly skin all over the body, which is very, very uncomfortable. if you didn't know. Off-target effects are when the CRISPR removes the wrong parts of DNA. It is uncertain what this could do to the human body, but mistakes like this are very possible and could be very detrimental effects on the human's compass, aka the DNA. CRISPR should not be used on human beings until the technology is completely flawless. Minor mistakes can have huge effects since DNA is the human's compass. We do support CRISPR and the study of it in the way that it can benefit many people's lives, but until this technology is sound, it should not be tested on humans.